Hello everyone, myself Divya Tiwari, and through this video, I'll be sharing the work I have done in this summer as the SRISHTI intern under the supervision of Dr. Kavita Vemuri ma'am. The title of my project is Microstate Analysis of Resting State EEG Data, where EEG stands for electroencephalography. Now, to understand the project, we first have to have a basic understanding of the brain signals. Now, the brain is the main organ of the human central nervous system, and it is made up of billions of cells called the neurons. Whenever we perform any activity or even while resting, our brain is active the whole time, and these neurons interact with each other via neurotransmitters. The synaptic transmission is triggered by the release of neurotransmitters, which causes a voltage change across the cell membrane. Any synaptic activity generates an electric field called postsynaptic potential lasting around 10 to hundreds of milliseconds. Now, by synaptic activity, I mean when a neurotransmitter is traveling from one neuron to another, it travels through the synaptic spaces present between the axon and dendrites of two cells. Now, this potential of a single neuron is too feeble to be noticed, and hence EEG captures the potential activity of a group of neurons. This is the reason why EEG has poor spatial resolution. The neuron oscillations that EEG can measure are visible in raw, unfiltered, and unprocessed data as well. However, the signal is always a mixture of several underlying base frequencies, which are considered to reflect certain cognitive, affective, or attentional states. The band frequencies are gamma, beta, alpha, theta, and delta waves. As you can see, if alpha wave is being seen in the EEG waves, that means that the person is in the meditation, creativity, or relaxation state. Now, electroencephalography or EEG is a non-invasive brain imaging technique. It is an exceptional tool for studying neurocognitive processes. It has a very high time resolution, but spatial resolution is not as good as fMRI. It was discovered by Sir Hans Berger, who is known as the father of EEG. It directly measures neural activity. It is inexpensive, lightweight, and portable. It records electrical activity and brain waves using electrodes placed on the skull. We'll talk about this in a later slide. Now, it is primarily the synchronized activity of pyramidal neurons in the cortical brain regions, which can be measured from the outside. These neurons are called pyramidal because their cell bodies are triangular or pyramidal in shape. They are oriented perpendicular to the cortical surface, and this unique orientation of cells generates a very stable electrical field. Cells in deeper brain structures do not have this specific orientation. In short, with EEG, we try to capture the electric signals generated by these pyramidal cells. And the electric signals that we capture are in the form of frequency bands with specific frequency ranges. Now, how the EEG data is collected. Recordings are done with electrode arrays placed on the skull, which capture the electrical activity and brain waves. The standard international system is called the 1020 system, where the electrodes are placed at specific locations in the cap, be it 32 electrodes, 64 electrodes, or the number may vary depending on the scope of the experiment. The recorded data is digitized and sent to an amplifier, and once the data is amplified, it is displayed as a time series of voltage values, as shown in the figure on right. Now, in this project, this is the workflow that I followed. The microstate analysis of 111 EEG is done using a novel approach where the EEGs are averaged into a single EEG data representing a healthy brain signal. For the project, firstly, some basic understanding of EEG, microstates, and all the necessary programming languages was acquired in the first 2 or 3 weeks. Now, once I had the basic understanding, I imported the data and did the necessary pre-processing. When I'm saying imported the data, it means the data that was used for this project was open source. Next up, topographic analysis, such as the global field power, was done, and then the microstate identification was done using different clustering algorithms. The two most important one being k-means clustering and modified k-means algorithms. Once the algorithms were applied and the microstate classes were obtained, characterization and analysis was done using statistical measures such as explained variance and microstate transition probabilities. In a typical EEG signal processing, these following steps are followed. When raw EEG data is obtained, it is pre-processed using several methods such as filtering, segmentation, artifacts removal, referencing, and resampling. Then, certain features are extracted depending on the scope and paradigm of the research. The popular ones being time domain features, frequency domain features, and statistical features. When these features are extracted, certain specific features are selected for further analysis, and classification or pattern recognition algorithms are applied on these selected features. The most popular ones being k-nearest neighbors, support vector machines, artificial neural network, random forest. This is for the typical EEG signal processing. Now, in the case of microstate analysis, classification unsupervised learning algorithms such as k-means clustering, modified k-means clustering, DV-scan are the popular ones. Once the algorithms are applied, the results are then visualized and interpreted. Now, in the current study, pre-processing pipeline included of these three steps. Firstly, bandpass filtering was done. A low-pass filter of 40 Hz and a high-pass filter of 0.1 Hz was applied. Hence, the resulting EEG signal contained frequency ranges between 0.1 to 40 Hz. Next, bad channel interpolation was done, where bad channels were reconstructed by interpolating their signals based on the signals of the good sensors around them using the spherical spline method. Lastly, independent component analysis was performed. It's a technique for estimating independent source signals from a set of recordings in which the source signals were mixed together in unknown ratios. It is the common example of blind source separation technique. Now, each EEG is separated into 20 independent components, and then through automatic labeling, signals from all sources other than the brain were rejected. Lastly, referencing was done. EEG measures a voltage between each electrode and a reference electrode. This means that whatever signal is present at the reference electrode is effectively subtracted from all the measurement electrodes. Hence, the average of all electrodes is set as a reference. Now, traditionally, researchers analyze EEG by characterizing the temporal waveform morphology and frequency distribution of recordings at specific pre-selected electrodes. A different approach that consists the information of all electrodes is to represent the EEG signal by the spatial configuration of the electric fields at the scalp that can be conceptualized as a topographical map of electric potential. Microstate analysis is an alternative EEG representation based on topographic analysis. Now, what are microstates? They are the quasi-stable states which remain stable for 80 to 120 milliseconds. Microstate analysis is a method where states are defined by topographies of electrical potentials over a multi-channel electrode array. The goal is to segment the recorded time series data into microstate classes so EEG samples that belong to the same class have as similar topographies as possible. Segmentation is done using topographical clustering methods. In the current study, 111 EEG data were used and averaged into a single EEG data representing a healthy brain signal, and then it is further used as an EEG signal of a single subject. The literature reviews and results of the current study suggested that clusters of 4 and 7 are the optimum number of clusters, and the microstate classes so obtained are in adherence to the previous studies made. Now, comparing different clustering algorithms on the given data set were compared to see their performances, and subject level analysis approach was used to obtain the microstates for the data. The most common clustering algorithms were k-means clustering algorithm and modified k-means clustering algorithm, which were compared in the present study. For the k-means clustering algorithm, I tried to use the elbow method and Silhouette method to find out the optimum number of clusters, but unfortunately, they didn't work out. Hence, modified k-means clustering was used for the further studies. The following 4 scores were used, which were based on the intercluster distance and intracluster distance. And as you can see in the figure on the left side, clusters number 4, 5, 6, 7, and 8 were giving good matrices for the following 4 scores. Hence, the clusters so obtained were compared for their global explained variance. These are the clusters obtained, and as you can see, as we increase the number of clusters, the global explained variance also increases. Hence, I did some literary review from the past studies done in the same paradigm, and depending on the scores obtained in the previous slide and the literary reviews, cluster number 4 with GEV 0.622 and cluster number 7 with GEV is equals to 0.686 comes out to be the most optimum number of clusters. Now, the typical softwares used for microstate analysis of EEG data is MATLAB and its extension EEG Lab Toolbox. However, in the current work, I used Python, which is an open source programming language having a rich set of inbuilt packages and modules. NumPy, MNEPython, and Microstate libraries of Python were extensively used in this project. With this, I would like to thank the SRISHTI team and my supervisor for providing me with this healthy opportunity. Thank you.